Would somebody confirm you can hear me?
I can hear you.
Thank you. Sorry for being a little late. Today, besides the normal 12 steps for logging in, I had to prove my existence and I had to go dig up my phone for Microsoft to call me. Before we begin, are there any questions? Let me shut some of these things down. Come on. All right. It's not scrolling to where I want to. Today is May 30th, and we are supposed to be finishing chapter 14 and beginning chapter 13. I'm not sure we'll get there. Hopefully we'll be close to finishing chapter 14. There is no lab today. You should be working on your unknown project. Any questions about the schedule or what we're doing? Did we even finish chapter 8? I don't remember. Let me check on that. It looks like we have not finished chapter 8. No, I did talk about genes and evolution. All right, so we did do that one. So let's close that down and begin chapter 14. When we started here, didn't we? All right, I've got marked that we were talking about the extent of host involvement in an infection, where the primary infection is your acute first infection, and it can weaken the body so that then you can have a secondary infection. Any question about any of that? Actually, we're pretty close to schedule, so we might finish this today. Now, let me see if I didn't lose it. So when we're talking about a disease, it's important to talk about the severity or duration of a disease. And some diseases have a latent period where we call the disease goes latent or latency, a disease where the patient has a period with no signs or symptoms. The pathogen is inactive. Often latent diseases are viral diseases, but I'm not sure that's true for all diseases. An example of a latent disease is when you have a cold sore. When you have the cold sore, the virus is actively replicating, causing disease, and then the cold sore goes away, and the virus is still there, but it's gone latent. And we call the disease is in its latent period, where you have no signs or symptoms, and actually the virus isn't replicating either when it's latent. And then the disease can leave latency where the virus begins replicating again, and you'll get another cold sore. A classic latent disease is chicken pox, where the child gets sick, gets chicken pox, and then the virus goes latent. It may go latent for decades. And then the virus comes out and replicates, causing disease again. And it's not the chicken pox. All later times it's called the shingles. Any question about any of that? Another concept you need to know about is herd immunity. When many immune people are in the population, some diseases cannot spread. Actually, all diseases, as long as you have the percentage high enough. Let me blow this up so I can show it better. And if enough people are immune, then the disease can't spread. We call that herd immunity, where the immune people are protecting the non-immune people. Like when a disease first hits and nobody has immunity, so everyone is susceptible, almost everybody in the population will get the disease, assuming the disease spreads. And they were actually afraid that COVID-19 would spread like this, which is why they took extreme measures to try and prevent the spread of COVID-19 when it first came to America. However, if enough people in the population are immune, then when somebody becomes sick, only people nearby those sick individuals will get the disease. The rest of the people who are not immune, meaning the blue people further away, will not get the disease because the disease has to go through this person or that person before it can get to this person. And these people are immune, so they're protecting this non-immune person. And this concept where the immune people are protecting the non-immune is called herd immunity. It's vital for protecting the population to have enough people immune to the disease so we get herd immunity. Now, with some diseases, herd immunity has to be very high, like, is it measles? I think it's measles. You need over 90% of the children to be vaccinated, or measles can spread to the non-immune people. With COVID-19, it's getting close to 90%. The last figure I read was 85%, but COVID-19 is proving to be better at spreading and spreading faster and spreading easier. And so it's probably over 85% now, meaning we have to have 85% of the population immune, or COVID-19 will continue spreading in the population. And then you'll never get rid of it. And guess what? That's what's happening, because we do not have 85% of the population immune to COVID-19. And we will probably never get 85% of the population immune to COVID-19. Surprisingly, with children, we do have usually about 90% of the population immune to measles. However, every once in a while, the immunity drops below 90, in which case we have a measles epidemic among children. And there was a case where measles were spreading at Disney because there were a large number of children altogether at Disney, and there were enough of them that were not immune that the disease was spreading. I don't remember if it was Disneyland or Disney World, but one of the Disney amusement parks in the United States was spreading measles. Not purposely, but just because children had it, went to Disneyland or Disney World, and the measles spread from them to other children. Any question about any of that? Let me zip up my coat here. I'm a little cool for some reason. Disease typically follow certain patterns. There's normally a specific sequence of events associated with an infection by a microbe and the subsequent development of disease. First, the disease organism has to come from a reservoir. So there has to be a place for the disease to be when it's not actively inside a person. The pathogen must be transmitted to a susceptible host. The pathogen has to invade the host, and the pathogen has to multiply. Furthermore, the pathogen has to injure the host. If you do not have injury of the host, you will not have signs or symptoms of disease. Now, it's possible that the disease does not injure the host and the patient has no signs or symptoms of disease, but the patient is asymptomatic, and then they can spread it to other people. The illness ends when one of two things happens. First, the host immune system destroys the pathogen, and then the pathogen is no longer in the host, causing disease. Or the other thing is well, I guess the pathogen could leave on its own. Sometimes they think human cold does that, although other people argue it's actually the immune system destroying the virus in the patient. But it's possible the illness could leave on its own. Or the third possibility is the illness kills the patient and then the disease ends, at least in that patient. Any question about any of that? All right. There are predisposing factors that can make the body more susceptible to, and therefore affect the occurrence of disease, making disease more likely to occur. For example, females have a shorter urethra than males, and females have a higher incidence of bladder infection because of that. There are inherited traits which make a person more predisposed to getting diseases. For example, patients who have the sickle cell gene, sickle cell anemia, are more likely to get most diseases than a normal individual in the population. The climate and weather can also predispose a person to getting disease. Everybody knows that colds and flus are more likely to spread in the winter. And there's actually several reasons for that. For one thing, people stay indoors, so they're closer to people. They have their windows shut or their doors shut, whatever. And that means the air in the room is more stale. Once it gets infected, it's more likely to stay infected. And so disease is more likely to spread in the wintertime, at least with colds and flus. But there's other factors as well. And that is the climate and weather can weaken the person's immune defenses, such as, I don't know, drying out the nasal mucosa, making it more likely to get sick. And then it's been discovered that the UV light of the winter is weaker. And so the colds and the flus, viruses, are less likely to be killed by light when somebody goes outside. Plus, when you do go outside, at least with the flu, it's well known that the cool temperature helps preserve the virus. And so the virus will live longer in the wintertime than it would in the summertime, just because of the temperature, as well as the fact that there's stronger UV light in the summer, and that would kill the virus as well. Fatigue predisposes people to getting diseases. And this should be a concern to you because students often are fatigued, either from studying too much, working too hard. Some of us teachers are slave drivers, or maybe some of you students just party all the time and you don't get enough sleep. Age is another factor affecting disease. Most diseases, let me see if I can show this here. I'm not getting paint. Let me do a search here. There it is. Most diseases affect the very young and the very old. So we have the incidence of disease here, and then just the time on the x-axis. The very young are more likely to get diseased and sick, and then the very old. And as you increase in age, your risk will decrease for the young, and then as your age increases for the old, your incidence of getting disease, as well as the severity of disease, increases. And that's true for most diseases like colds and flus and I don't know, most other diseases. Usually in elderly childhood and the teenage years are someplace around here. And then young adults are the population that are less likely to get diseased. And then middle age starts climbing, old age climbing bunch. The very elderly have the highest risk of getting disease. That's not true for COVID-19. Let me make another graph here. For COVID-19, only the very young are susceptible. I'm talking about newborns and very, very young, like the first couple of weeks of life. And then it drops very low in childhood, even early childhood. And then it climbs very rapidly, very great risk at elderly population. So COVID-19 is unusual in that the elderly have a much greater risk than the young, with the exception of newborns, or close to it. Newborns do have greater risk than the majority of the population. Any question about that? COVID-19 is unusual in that it's not a symmetrical curve where the young and the elderly are at similar risk. And then, I don't know, this is young childhood here. They're also at risk. Like a young child is much more likely to get an ear infection than an adult or even a teenager. But COVID-19, like I said, this only affects generally the first few weeks of infancy. Lifestyle can predispose an individual to getting disease. It's well known that smokers are much more likely to get a respiratory disease than non-smokers. And that's because smoking damages the mucosal membrane, as well as it probably weakens the host immune system. So you're more likely to get the disease because your immune system's a little weaker. But it's mostly because the mucous membrane is damaged and then the infection can get an early start. Whereas in a normal person, the infection has to get through the mucosal membrane before they get disease. And then chemotherapy can predispose an individual from getting diseases. For one thing, chemotherapy tends to weaken the host immune response, and so they're more likely to get disease. Any questions about any of that? Disease tends to follow a specific pattern of development. So once the host defenses are breached, disease develop according to a certain sequence. This sequence tends to be similar for acute and chronic diseases, meaning almost all diseases follow this pattern of development. First, there's an incubation period. The incubation period is when the patient is first exposed to the agent causing disease till the time when the patient first has signs or symptoms of disease. Any question about that? And on this graph here, we have the number of microbes increase on the y-axis, time on the x-axis, and then the Also on the y-axis is the degree of sickness, with higher being more sick, lower being less sick. So yes, usually the number of microbes increases the sicker a patient gets. Any question about that? It isn't always true. I'll talk about an exception in just a little bit. But after the incubation period, when the patient first has signs or symptoms, we call it the prodromal period. This is the period when the patient is first becoming sick and they have mild signs and symptoms. Like when you're first coming down with a sore throat, you say, geez, am I getting a sore throat? And you have to think about it. And you say, yeah, I'm coming down with a sore throat. That's the prodromal period. The prodromal period ends when the patient's signs and symptoms are no longer mild, when you have full-blown disease or signs and symptoms of disease. The patient is usually the most sick in the period of illness. In the period of illness, you also have the most organisms causing disease. In the period of illness, you don't have to think about whether you have a sore throat or not, because your throat is burning and it's quite clear to you you've got a sore throat. When the patient starts getting better, we call it the period of decline. One example, which is a clear distinction between the period of illness and the period of decline, is when a patient has a fever and then the fever breaks. That begins the period of decline. In the period of decline, the patient still has organisms causing disease. They still have signs and symptoms of disease, but the signs and symptoms are getting better. The number of organisms is decreasing. The period of decline is followed by the period of convalescence. This is when the number of disease-carrying organisms goes below a threshold, and now the patient has no signs or symptoms of disease, but you notice they still have the disease-carrying organism. And that's the start of the period of convalescence. The period of convalescence ends when the patient no longer has any disease-causing organisms in them. Any questions about the stages of the development of disease? Okay, here's a question for you. During which period is a patient unlikely to spread disease? I shouldn't say unlikely. At what period of disease is a patient unable to spread disease? Anyone have a guess? If no one guesses, I'm going to make this a quiz question.
Incubation.
That's a good guess, but actually that's wrong. Anyone else?
Period of covalences?
Sorry again?
Period of covalences?
You mean the period of convalescence?
Yes.
That's also a good guess because you have no signs or symptoms of disease. However, this is a trick question. In the different periods, you always have disease-carrying organisms. So at every period of disease development, you can spread disease, including in incubation and the period of convalescence. Now, that doesn't mean that you're likely to spread it, but it does mean that you can spread it. In fact, for many common diseases like colds and flus, you are most likely to spread it, or at least an adult is most likely to spread disease late in the incubation period before they have any signs or symptoms, or during the prodromal period. And there's less organisms in these two periods than in the period of illness. What's happening in the period of illness, at least for colds and flus, is in an adult, their immune system is responding to the disease, and the immune system is preventing the disease organism from leaving their body and then causing the illness to spread. I think this goes back to our caveman ancestors, and that is the adults were trying to protect their children from getting their disease. And that's why colds and flus are much more likely to spread late in the incubation period and in the prodromal period than in the period of illness. Now, if you're curious, I said it happens in adults. Actually, in children, that is not the case. Children do not appear to be concerned about spreading the disease to their parents, meaning their immune system is not responding to the disease in the period of illness and then decreasing the chance that it can spread. Any question about any of that? Here's another question for you, and that is, what period of illness do we sometimes not see in a disease?
Incubation.
No, you always have incubation period, because the incubation period is when you first are exposed to the disease, and then before you have any signs or symptoms. And the reason why you don't have any signs or symptoms is initially, when the disease-carrying organism is in the patient, there's too few bugs, microbes, for the disease carrying organisms cause signs or symptoms of disease, meaning you need the number to increase above a threshold before you get any signs or symptoms. So that was a good guess, but no, you always have the incubation period. Another one you almost always have is a period of illness. Okay, so those two you always find. And that
So that
means,
wouldn't be the
obviously, I skipped
period.
one. So the prodromal period is one where sometimes you do not have it. And that is, sometimes I'm walking around fine, and the next moment it feels like somebody hit me over the head with a sledgehammer. Sometimes the flu is that way, where you're in the incubation period, you have no signs or symptoms, there is no prodromal period, and the next thing you notice, it feels like somebody hit you over the head with a sledgehammer. And that would be some cases of the flu, where you don't have a prodromal period. Another period that you do not have is the period of decline and the period of convalescence. HIV initially causing AIDS got the patient in the period of illness, and then the patients died. So there was no period of decline or period of convalescence. That isn't true today with modern medicine. They can keep a HIV patient alive for almost their normal lifespan. It's a little bit less than the normal lifespan, but they can keep them alive for a long period of time. Usually, these patients never develop AIDS. And if they do develop AIDS with medication, you can sometimes bring them out of AIDS. So you move from the period of illness to the period of decline and period of convalescence. Now, with HIV, the organism never goes away. It is true there might be a few patients where they wonder if the organism ever went away, but they never proved it, because HIV can be hiding in your DNA in one cell, and then it could always come out later. But there were a few patients that wondered if the HIV actually left the patient, meaning that they didn't have any signs of HIV. Any questions about the period of the development of disease? Let me end the show here and make sure I've got everything I'm talking about. All right. Oh, I would never ask a trick question like during what period of disease are you unlikely to spread disease? I would never ask a trick question like that in a quiz. I should mention that my mother always told me, "Oh, I'm getting better now. I'm no longer contagious." And I would always argue, "Mother, you're talking to a microbiologist. I know better." Not until you.
I have a question, Professor.
Uh-huh.
Can we expect to see that picture on one of the like a final? Will we need to know that chart for the final?
You should know this chart for the final. I'm not likely to give you the chart for the final,
Okay, thank you.
but I'm likely to ask a question about the development of disease.
Okay, thank you.
All right. All right, let's talk about the spread of infection. Disease has to come from a reservoir of infection. This is a source. Reservoirs are continual sources of disease. Reservoirs can be living or non-living. A very big reservoir of human disease are other humans. So we call them human reservoirs. For some diseases like AIDS and gonorrhea, humans are the only reservoir of these diseases. And it is true that they think HIV originally came from, I think it was from a gorilla, not a gorilla, a chimpanzee. But generally, HIV comes only from humans. Okay. There can be carriers. So some humans can be carriers. They have an apparent infection or a latent disease, and they can spread it to other people. This is particularly true of young people in COVID-19, where it's estimated about 50% of children, teenagers, and young adults will be asymptomatic for COVID-19. There can be animal reservoirs where diseases can come from animals. Some diseases, like rabies, only come from animals. Lyme disease also only comes from animals, meaning you need a tick to bite you before you can get Lyme disease, or you need an animal to bite or scratch you to get rabies. Rabies and Lyme disease do not spread from person to person. Some diseases called zoonoses are animal diseases that may be transmitted to humans. And then there are non-living reservoirs. For example, botulism, tetanus are examples of diseases that usually come from a non-living reservoir. Botulism from canned food, and it's canned so the food is no longer alive. Tetanus, usually from a puncture wound. So the nail or the glass or something. Some common non-living reservoir includes soil, water, and then lettuce, and it's non-living because it's been picked. Many cases of gastroenteritis come from lettuce or something similar like that, like what do you call that? Cabbage, where people eat it without cooking it and then they get a disease. Any question about reservoirs? Disease has to be transmitted, and there are three principal routes by which the causative agent of disease that can be transmitted from the reservoir of infection to a susceptible host. There's contact transmission, such as direct contact to contact, handshake, kissing. Vehicle transmission, where some object picks up the disease organism from an infected person or agent, and then the object gives it to a healthy patient, like a cup. And then there's vector transmission, and that's where a animal gives the disease to a healthy person, like when a mosquito bites an infected person, and then the disease is picked up by the mosquito, and then the mosquito bites a healthy individual, giving the disease to the healthy person. Any question about any of that? All right, let's talk about contact transmission in a little more detail. There are 3 types of contact transmission. There's direct. This is person-to-person transmission, requiring close association between the infected individual and a susceptible host. Kissing is an example of direct person-to-person transmission. A handshake, another case of direct person-to-person transmission. Indirect transmission is where the disease agent spreads from the reservoir to a susceptible host via a non-living object. We call non-living objects fomites. For example, somebody sneezes on their hand, causing the disease-carrying organism to get on their hand. They then touch money. The disease-carrying organism gets from their hands onto the money, and then the money spreads it to a susceptible patient. Another case of indirect contact transmission would be a baseball, where many children touch the baseball and one of them carries the disease-carrying organism onto the baseball, and then everybody else who touches the baseball after that is potentially exposed to that disease-carrying organism. Disease-causing organism, sorry. And then there's droplet transmission. That's where transmission via an airborne droplet travels less than 1 meter. I'll talk a little bit more about that. Here we're looking at direct contact transmission, indirect contact transmission being the baseball, and then droplet transmission when somebody sneezes. If you're within 1 meter, about right here, that they call droplet transmission. Any question about that? I don't like the droplet transmission because it's contact transmission, and we have vehicle transmission. There's one type, airborne transmission. Airborne transmission is when the sneeze or cough travels more than 1 meter, and you're getting it still from the same sneeze or cough. But like I said, if it travels 1 meter, they call it droplet transmission. And if it's over 1 meter, they call it airborne transmission. And droplet transmission is contact transmission. Airborne is vehicle transmission. And I just decided this is a little bit arbitrary, so I'm not going to quiz you on that. Okay? Because to me, that's a sneeze. It doesn't matter where you get it. If you're within 1 meter or beyond 1 meter, you still get it from that sneeze. Any question about that? All right. So vehicle transmission is usually when transmission occurs by an inanimate reservoir, such as a fomite, but it can be spread by food, water, and body fluids. And there's 3 types of vehicle transmission. Waterborne transmission, such as when the disease-carrying organism contaminates the water, such as when sewage contaminates drinking water. That doesn't happen very much in the first world countries, but it actually is quite common in third world countries. Foodborne transmission is where somebody who's sick puts the disease-carrying organism on the food, and then the patient, the healthy patient, eats the food, and they get the foodborne transmission. Foodborne transmission can occur when the food is undercooked, poorly stored, or unsanitarily handled. Like people who are handling food, selling in a restaurant, they're supposed to be wearing plastic gloves to reduce the transmission of foodborne transmission. And then there's airborne transmission, which I've already talked about. That's when a person sneezes or coughs and it goes beyond 1 meter, and then somebody breathes that in, or it gets on their eyeball, or I guess if they breathe it in, either the mouth or the nose, and they get sick. Any question about vehicle transmission? There's also vector transmission, and vector transmission happens when an animal carries the disease-causing organism from a reservoir to a susceptible host. The most important group of vectors are the biting arthropods, especially fleas, ticks, and mosquitoes. Vectors can transmit disease in one of two ways. There's mechanical transmission. This is passive transport of the disease-carrying organism, such as this fly is passively transporting the disease-carrying organism by picking it up on its feet, their leg, and then the fly lands on the food, passively putting the disease-carrying organism onto the food, and then the food is eaten by the susceptible patient. Any questions about mechanical transmission? Biological transmission is where the pathogen actually reproduces inside the vector. The best known case, or one of the easy known cases, is the mosquito. The malaria parasite actually reproduces within the mosquito. And then when the mosquito bites in a susceptible host, they can transmit malaria. Any question about that? All right, let's move on to another topic, and that's nosocomial infections. This is one of the most important topics you can learn in microbiology. And that's because most of you are going to find work in a healthcare setting, meaning either a doctor's office or a hospital or a nursing home or something like that. And a nosocomial infection is an infection that the patient did not have before they got inside the healthcare setting. And then they got the nosocomial infection from their stay in the hospital-like or the healthcare setting. Any question about that? And why that's a concern to you is you don't want to spread around nosocomial infections. You don't want healthy patients, or at least somewhat healthy patients, picking up a nosocomial infection just from checking into a healthcare setting. Nosocomial infections, also called hospital-acquired infections, are infections that result as a result of a stay in a healthcare setting. The disease-carrying organism was not present or incubating at the time of admission, but the patient picked it up in the healthcare setting. It's estimated that 5 to 15% of all hospital patients acquire a nosocomial infection each year in the United States. This is about 2 million people per year, or 20,000 deaths from a nosocomial infection each year in the United States alone. It used to be the 8th leading cause of death in the United States. Because of COVID-19, this has probably been bumped to the 9th leading cause of death now. Any questions about nosocomial infections? So about 1 in 10 hospital patients gets a nosocomial infection, and this is from the CDC. About 50% of nosocomial infections are preventable, and this was done from a study in 2013. Zimlickman's 2013. You don't need to know the cost of nosocomial infection, but you should know that it's a real big cost. Nosocomial infections result from the interaction of 3 factors that occur in the healthcare setting. First, microorganisms are present in the healthcare setting. Like when a patient checks into the hospital, there's microbes all over the hospital. Hospitals are a major reservoir of various pathogens because when a patient gets sick, if it's a pathogen causing it, they will be checked into the hospital, and there will be a source of a potential nosocomial infection. Some normal microbiota are opportunistic pathogens, and they can be nosocomial infections. And then some nosocomial pathogens are antibiotic resistant, and they're a real problem with a nosocomial infection because let's say the patient comes down with a nosocomial infection, the doctor prescribes the normal antibiotic, and then it doesn't work. And so the doctor scrambles and runs a test to determine what antibiotic to use, and he goes to the second antibiotic, and the pathogen's resistant to that antibiotic as well. The test comes back and it says, oh, there might be this antibiotic that works, but by this time, the patient has already died of the nosocomial infection. And then, of course, there are microbes in the air and on the floor and all over the hospital. It's unavoidable. Even flower vases have microbes that can cause a nosocomial infection. Pseudomonas aeruginosa is a nosocomial infection, and it lives anywhere where there is moisture. So it can be in a flower vase. Another reason why nosocomial infection occurs in the healthcare setting is the patient is oftentimes compromised when they check into the healthcare setting, such as the hospital. Their resistance to infection is impaired by disease, therapy, or burns. Broken skin or mucous membrane compromises the host, makes infection more likely. Burns, injections, ventilators, urinary catheters, even the administration of anesthesia make the patient more susceptible to getting a nosocomial infection. And then the immune system can be suppressed for the patient who's checked into the hospital. The suppression could be from drugs, radiation therapy, burns. All of these weaken the immune system. If the immune system is weak, you're more likely to come down with a nosocomial infection or any other type of infection. And then there's a chain of transmission of the nosocomial agent inside the hospital or healthcare setting. There can be direct contact transmission, and that could be as simple as patient to patient. But it could be picked up by the hospital staff from the patient, and then the hospital staff spreads it to other hospital patients. This is a real concern for special care units, and they usually take steps to prevent the transmission of disease in a special care unit. Hospital staff are supposed to wash their hands or wear gloves and take off their gloves after they touch a patient before they go on to the next patient. There can be indirect contact transmission as well, and this occurs via a fomite. Urinary and IV catheters, contaminated surgical dressing can all spread transmission indirectly. If you ever look at one of the old medical films, like a hospital setting film or something in the 1960s, the doctors proudly wore their stethoscope around their neck, and they'd take it and then they would touch the patient's skin with the stethoscope. And then when they were done, they would just drop it and it would thump against their chest. And then they'd go on to the next patient and use the same stethoscope to touch the other patient's skin. The stethoscope in the 1960s was actually spreading around nosocomial infections. Nowadays, doctors know that if they do touch skin with their stethoscope, they're supposed to sterilize their stethoscope, or at least clean it, between patients. And that's indirect contact transmission. And then there's also vehicle airborne transmission, where the ventilation system can actually spread disease-carrying organism. Obviously, colds and flus can be spread by the ventilation system in a healthcare setting, such as a hospital. I remember when the first patients came to the United States with Ebola, and there was real concern that when they were treating the patients in the United States with Ebola, that it might spread by the ventilation system. And I think they took steps to control that, meaning they would filter the air that was being taken in by the room of an Ebola patient, just to make sure that Ebola wasn't spreading by the air in the hospital. Any question about the chain of transmission? All right, so those are the three factors of why nosocomial infections occur in the hospital. There's microorganisms in the hospital environment. The patients usually are compromised when they check into the hospital. And then there's a chain of transmission within the hospital. So how do we control nosocomial infections? Well, there's certain procedures which are generally implemented to control the spread of nosocomial infections. You have learned aseptic technique now in microbiology. Whenever you are employed by a healthcare facility, you should always use aseptic technique, and then you will be less likely to spread nosocomial infections. You should use proper handling when you're handling contaminated materials. And then when you're done handling that material, you should wash your hands or take off the gloves. Frequent and thorough hand washing have actually been shown to reduce the spread of nosocomial infections by 50%. So you should have frequent and thorough hand washing. And then once you touch a patient, you should go wash your hands. And then there's education of the staff, patients, and visitors. Like the doctors started learning about nosocomial infections in this country sometime in the 1960s. In the 1970s, they started educating the staff in the hospitals. In the 1980s, they started educating the patients and the visitors. Now, whenever you go into a hospital, you'll see a sign outside the patient's door or in the elevator saying, stop the spread of nosocomial infections, sanitize your hands. And usually outside the door of each hospital, there's sanitizer, as well as usually in the elevators, so that people can sanitize their hands before they go and see a patient and then potentially spread a nosocomial infection to them. So the point is, even the patients and the visitors now are educated about controlling nosocomial infections. And actually, it's worked. In the last 5 years, the number of nosocomial infections in the United States has dropped by about half, meaning 50% of them. So we're getting better at controlling nosocomial infections. There are some other steps you can use to control nosocomial infection, and you can check the online chapter notes to learn about different things you can use to prevent the spread of nosocomial infections. Any question about any of that? All right, if not, let's change topics a little bit and talk about epidemiology. Epidemiology is the study of where and when disease occurs and how diseases are transmitted in populations. The CDC collects all this information in the United States, analyzes it, and then publishes it in the Morbidity and Mortality Weekly Report, which is usually abbreviated MMWR, and you can find that at www.cdc.gov. Let me see if I can open that. The trouble is it's difficult to find the link for MMWR now. It used to be very easy to find. You just scroll to the bottom of the page. I might have to do a search now. Anyone see a search page? Oh, here we go. I'm not sure this will be it. This is specifically diseases. Oh, there it is. Morbidity, Mortality Weekly Report. Maybe I'm not seeing what I'm looking for. I'm not sure. These are specific agents. I wanted all of, oh, there it is. There we go. There's a chart. So these are diseases of Western virus, different viruses. There should be, there it is here. No, that's still only one disease. Different states. Well, I'm not finding exactly what I was looking for, but that's okay. Anyways, there's the MMWR, and it did show some diseases. Where is it? Let me blow that up. So different viruses of encephalitis. I don't know this one. I've never heard that one. West Nile virus, St. Louis encephalitis, lacrosse, and Jamestown Canyon virus. There are notifiable diseases. These are diseases that physicians are required by law to report. Normally, a physician will report this to the county health authorities, which for Vancouver would be Clark County. So they report it wherever the health facility is for Clark County, probably in Vancouver. I used to live in Washington County, and so you report it to the Washington County authorities, and that was in Hillsborough. And then the county authorities send it to the state authority, which for Oregon, that was Salem. I'm not sure where the state health office is in the state of Washington. It might be Olympia, but I'm not sure, wherever it is. And then the state will report it to the Center for Disease Control, and that would be Atlanta, Georgia, and they collect it all, compile it, and then publish it weekly, as well as make an analysis of it. And they publish it weekly in the mortality and weekly report, morbidity and mortality weekly report. Any question about any of that? Some terms of epidemiology. Morbidity is the incidence of a specific notifiable disease. The morbidity rate is the number of people affected in relation to the total population in a given period of time. The mortality is the deaths from a notifiable disease. The mortality rate, the number of deaths from a disease in relation to the population in a given time. Any question about any of that? Any questions about chapter 14? If not, we're just going to get started with chapter 13. Let me shut that down. Chapter 13 is going to be studying viruses, viroids, and prions. Viruses, viroids, and prions. So as is normal, I show you a slide detailing the major goals and a brief outline. So you should always know the terms. What's a virus capsid, a virus envelope, virus spike? What's a bacteriophage? What's a lytic virus, a lysogenic virus? What's a prophage? What's a provirus? What is a latent and persistent viral infection? What is a prion? And what is a viroid? 2, understand the characteristics the viruses have, their structure, and their classification. 3, understand about viral multiplication, meaning viruses multiply in one of a few different ways, and we call these viral life cycles. For bacteria viruses, they only replicate in 2 ways. For animal viruses, they replicate in about 4 or 5 ways. We'll talk about at least some of them. And then 4, understand what are prions and viroids. A virus, it has only one goal, and that's to replicate. How do they do that? Viruses generally invade a host cell, hijack it, and then force that host cell to make viral copies, which then escape from the cell to spread the virus. We have a little video here on viral replication. A phage is a bacterial virus. We'll talk about that later.
Is about to take place. Deep bacteriophages. Spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
All right, any questions about that video? Viruses are generally inert when they're not within a host cell. In fact, they display little or no metabolic activity outside of their host cell. So outside of their host cell, viruses have little or no metabolic activity. Viruses require a living host cell to multiply. So we can say viruses are obligate intracellular parasites. They are a parasite of a cell, and they have to replicate within that cell while they're an obligate intracellular parasite. They multiply within living cells by using the host cell's enzymes, host cell's nucleic acid, host cell's amino acids, and the host cell's ATP. In fact, viruses have no ATP generating mechanism. They rely on their host cell to obtain the ATP that they need. Viruses do have a genome, and that genome contains either DNA or RNA, but never both. And yes, viruses can have an RNA genome. If you don't know, our cells only have DNA genomes. But a virus can have either DNA or an RNA genome. Viruses are also surrounded by a protein coat that surrounds the nucleic acid. So here we have a virus with this edge of it cut out. You can see the nucleic acid, the innermost molecule of the virus. And surrounding it is a protein coat. Some viruses also have outside of the protein coat a lipid envelope. So it would be about this area here. This virus does not have a lipid envelope. Any question about any of that? And viruses can be one of two ways. It can be with the lipid envelope or without the lipid envelope. And it just depends on the species of virus. Viruses have a host range. This is the range of host cells a virus can infect and then multiply in. The host range is determined by specific host attachment sites, meaning you need certain molecules on the cell wall, the flagella, or other molecules on the host cell that the virus attaches to. And these we call receptors, the molecules on the host cell that the virus will bind to. You may need cellular factors within the cell necessary for viral multiplication. Like all viruses need ATP, you need ATP within the cell in order for the virus to multiply. The virus itself has molecules on its surface that bind to the receptor. And these molecules, we either call them ligands or adhesions. Let me spell that out for you. So the molecules of a virus that bind to the host cell receptor are either called ligands or adhesions. These are two words that essentially mean the same thing. Adhesions named after, this is what the virus adheses to, and I'm not sure where the name ligand comes from. Most viruses only infect the cells of one or a few species. And usually they only affect certain cells of that one or a few species. For example, HIV only infects T cells, macrophages, brain cells, and maybe a few other cells. Most cells, HIV will not infect. It will not infect skin cells, will not infect muscle cells. I don't think it infects blood cells either. Well, obviously white blood cells, it can infect macrophages. But red blood cells, I don't believe it infects red blood cells. So most viruses are very specific and they only infect a few different cell types of one or a few species. For example, HIV only infects those cells of humans or one of the chimpanzees. There are two chimp species and only one of them HIV can infect. If you don't know, HIV can infect and can grow in a chimpanzee. However, it does not cause AIDS in the chimp. The chimp just gets a viral infection. It does not destroy the chimp's immune system. Any question about any of that? All right, as I mentioned, a bacterial virus is called the bacteriophage, and that could be pronounced phage. And sometimes that's shortened to phage or phages. So whenever you see these words, we're talking about bacterial viruses. I will either say bacterial viruses or bacteriophages. Viruses have a relative size, and their size is about 1/10 the size of a prokaryote cell. So obviously here we're seeing the size of a red blood cell, and obviously we're not seeing most of it. Right there is E. coli. E. coli is a little bit larger than your average bacteria, just a little. And that's because it has a longer length than most bacteria. Or you should say your average bacteria. Viruses come in different sizes. They range from 20 nanometers to 440 nanometers in diameter. But by their length, they range from 20 nanometers to almost 1,000 nanometers long. And yeah, they can be quite long, like Ebola is a very long virus, but it's very skinny. So Ebola is not the largest virus by the diameter. The largest virus by the diameter that we're going to talk about is megavirus. And it's a very, I don't know, spherical virus. Whereas Ebola has a smaller diameter, but a longer length. And those are the largest viruses. The smallest virus you know is poliovirus. at around 30 nanometers. There are some viruses, a few, smaller than poliovirus, but I doubt anyone's ever heard of them. Like bacteriophage MS2 is smaller than poliovirus, but nobody's probably heard of bacteriophage MS2. Any question about any of that? All right. When we're talking about virus, it's important to realize that the virus has a structure. A virion is a single mature, complete infectious virus particle. So usually when we're talking about a virus, we're talking about a virion. Viruses can have a genome, which is nucleic acid, and their genome can be DNA or it can be RNA. The genome can be double-stranded or single-stranded. So there's double-stranded DNA genomes in viruses, and that's not unheard of. That's what most of our DNA is most of the time, a double-strand DNA. Some viral genomes are single-strand DNA, and that's not unheard of in our cells either, because whenever our DNA is replicating, or whenever our DNA is transcribing, it is single-strand DNA. Some viral genomes can be double-strand RNA, and that's unheard of in our cells. Our cells do not have double-strand RNA. And then some viral genomes can be single-strand RNA. When we're talking about a single-strand RNA virus, it's important to state whether it is a positive single-strand RNA virus or a negative single-strand RNA virus. A positive single-strand RNA virus is one that can be read by the ribosome, so it will have AUG on it. The negative single-strand RNA virus cannot be read by the ribosome, and it will not have AUG. It'll have the corollary to AUG, which is UAC. You don't need to know that, but viruses can be linear or circular. And as you know, eukaryote DNA is linear, but prokaryote DNA is circular. So viruses can be either linear or circular. Some viruses, like the influenza virus, can have their viral genome in more than one segment. So there's several segments to the viral genome. And that's not unheard of in our cells because our genome is in different segments, and we call each segment a chromosome. Now, viruses, we don't usually call the segment a chromosome. For one thing, the viral genome is usually not associated with protein the way a eukaryote or even a prokaryote genome is. All right, any question about any of this? If not, I'm going to stop here, and I will be in the lab for 15 minutes to answer questions, but after 15 minutes, I will shut down the Zoom session, unless the questions go beyond 15 minutes. So you only need to show up in the lab if you have questions. Any questions? All right, I'll say goodbye.
Thank you.
All right.